thank you for being here. In the interest of time, I'm just going to jump right in. So I say until it doesn't go to the next slide. Okay, so as we all know, pollination is an essential ecosystem service to humanity. So it enhances plant genetic diversity and improves the quality and quantity of many food crops, and it forms a resource base for wildlife. So pollinator importance likely increases at low latitudes because there are more crop species and subsistence farmers are more dependent on these free pollination services. Pollination services in tropical montane ecosystems are potentially more sensitive to climate change than lowland or temperate systems, as they consist of a higher proportion of functionally specialized interactions. These pollination service decline can lead to decreased food provisioning and long-term decreases in global plant diversity. Next slide. Okay. In the mountainous areas of the tropics, it's important to know how bee richness and abundance varies with elevation, because our ability to predict bee response to climate change in tropical mountains depends on what we know of their altitudinal distribution. Species that are common only in a narrow portion of their full altitudinal range should be just as vulnerable to climate change as species with narrow altitudinal ranges. And understanding the abundance patterns of tropical montane bees also has many direct applications for tropical plant species predictions. Hotspots of bee diversity have been shown at mid-latitudes and arid environments. So since bees don't appear to follow typical global distribution patterns, their distribution along tropical mountains may also reveal unique patterns. Climate change in the tropics is predicted to reveal frequent and more severe droughts, as well as erratic precipitation, and to have a greater impact on tropical species, particularly ectotherms, species residing in the Pacific slopes of Mesoamerica, and mutualists. So the bees of Pacific Central America are among the most at-risk taxa. Here, we surveyed wild bee communities in two life zones. One was a lower zone consisting of tropical dry forest, and then an upper zone consisting of tropical pre-montane forests. And we did this to ask if bee abundance differs by elevation, what annual and seasonal changes in abundance are observed, and how does the relative abundance of bee species visiting the flowers of two crop species differ by elevation. So we established 3 spatially independent replicate elevational transects in the San Luis Valley of Monteverde in Costa Rica. Each of these 3 transects were separated by 1 kilometer, with the lowest elevation of each at around 800 meters above sea level, excuse me, and the highest at around 1,100 meters above sea level. The San Luis Valley is located on the Pacific slope of the Tuolumne mountain range. The landscape's typical of the tropical countryside, so some smallholder farms and forest remnants all kind of scattered throughout. There are very distinct wet and dry seasons. The wet season lasts from about May to November, and then the dry season lasts from around December to April. Within the study area, we also collected data from 15 smallholder farms and residential properties. So 5 of these sites contained cucurbita, 5 contained Persea americana, and then 5 contained both focal crop species, which totaled to 10 sites per crop species. So we focused on species belonging to 2 bee tribes, Ceratinine and Meliponine, and then we also focused on Apis mellifera. Species in the tribe Ceratinine are highly diverse and globally distributed, and there are about 15 known species to occur in Costa Rica. And the tribe Meliponine consists of several hundred described species, about 50 of which occur in Costa Rica. And then Meliponine species are generally distributed in the tropics and subtropics, but they do extend a little bit into the temperate area of the southern hemisphere. And then Apis mellifera is the only species belonging to the tribe Apini in our study area, and they are considered to be a super generalist forager. Persea americana, or avocado, one of our focal crop species, it's native to Mexico, Central and South America. However, it is cultivated in many different climates globally. So in Mexico, Meliponine bees have displayed a preference for avocado flowers, while Apis mellifera will actually forego avocado for more rewarding flowers. Most species in the genus Cucurbita are native to Mesoamerica. Species in Peponapus and Xenoglossa and the tribe Eucerini are obligate pollinators, so those squash bees. However, honeybees and stingless bees are occasionally found to be important as well. So we sampled along the 3 transects at 800, 900, 1000, and 1100 meters in elevation during June, July, and December of 2016 through 2019. So in 2016 to 2018, at each of the 4 sampled elevations on each transect, we placed 30 pan traps linearly along the transect. We hung 1 yellow and 1 blue vein trap. And then we observed all flowering plants along each transect for flower visitors, collecting bees using hand nets or jars. And then in 2018 and 2019, we used a honey spray solution in order to attract and capture bees in the tribe Meliponine. So for that, we just locate some broad vegetation in an open area and spray that with a honey water solution. And then lastly, we conducted 30-minute flower observations along 1 transect in 2017 and 2019, as well as within the 15 focal crop species sites during 2018 and 2019. We
use one-year mixed models to determine the significance of elevation and bee species abundances for bees collected in pan trap, vein trap, and 30-minute flower observations. And then Ceratinine species abundances were also analyzed to determine the significance of season on bee species or on bee abundances. Honey spray sample data were used to quantify the significance of elevation and the presence and absence of bee species from the tribe Meliponine. And then data from the 30-minute timed flower observations were used to quantify differences in the presence and absence of all the bee species across their elevation gradient. So, and then to evaluate how bee community composition differed across the gradient, we conducted a CCA ordination. And lastly, we calculated the similarity indices in order to quantify similarities in the bee community visiting squash and avocado flowers. Pan trap, vein trap, and 30-minute flower observations combined collected sufficient numbers of, unfortunately, only 3 bee species in our local tribes, and those 3 were Apis mellifera, Ceratinine rectangulifera, and Ceratinine trimaculata. So although the abundance of Apis mellifera was not significantly different across the elevations, the abundance of both Ceratinine species did significantly differ across the elevations. So the abundance of Ceratinine rectangulifera was lowest at 1100 meters compared to the other 3 elevations, while Ceratinine trimaculata had a significantly higher abundance at 1000 and 1100 meters compared to the 800 meter elevation. Season of collection also had a significant effect on Ceratinine abundance in pan traps. So on average, fewer individuals were collected during December samples, which are shown here, compared to the June and July samples. Of the 10 Meliponine species collected sufficiently during honey sprayed vegetation sampling, the presence of 5 significantly differed across elevations. And of the 21 species collected sufficiently during timed flower observations, the presence of 15 of these species significantly differed across the elevation gradient. A total of 5,678 bees belonging to 24 species were used in our constrained ordination. So the effects of elevation and transect ID together explained almost 42% of the variation in the bee community composition across their gradient. And elevation and transect ID each alone actually explained about 21% of the variation. The bee community visiting avocado flowers differed across the gradient, with the community present at 1000 meters and 1100 meters exhibiting the highest similarity, while the communities at 700 and 1100 meters in elevation were the most dissimilar. However, it is important to note that the species accumulation curve for the sampling did indicate that more sampling was needed here. For squash, the bee communities at 900 and 1100 meters were the most similar, while the bee communities at 800 and 1100 meters, as well as 800 and 900 meters, were the most dissimilar. So in summation, of 26 species, the presence of 15 were significantly different by elevation. The presence of 6 were not significantly different by elevation, and 5 species were actually collected too infrequently to statistically estimate their differences. So our results suggest that a combination of abiotic and biotic factors might account for the observed patterns. So for example, the 2 species that we most commonly collected in pan traps, that's Ceratinine rectangulifera and trimaculata, they were observed at all elevations across the gradient studied. But Ceratinine rectangulifera was observed more frequently in the lower zone,
while Ceratinine and Meliponine was observed more frequently in the upper zone. So these two species are nearly identical in morphology, nesting, and floral preferences, which suggests that spatial niche partitioning could be occurring here, potentially as a result of interspecific interference competition. Precipitation patterns and rainfall gradients could also have an effect on bee assemblages and distribution patterns. So Ceratinine rectangulifera and Trimaculata abundances appear to exhibit this interannual fluctuation that reflected the seasonal rainfall patterns, with low abundances observed during that dry season and then high abundances during the wet season. However, these fluctuations could also be due to seasonality and reproduction, as seasonal hibernacula have been observed in the area for these species. Our crop visitation data showed high turnover between some elevations among the crop visiting bee community, indicating the importance of elevation in determining these pollinator assemblages. Studies from temperate mountain-dwelling pollinator communities have predicted that species will expand their ranges at that cool upper elevational limit and then contract their ranges at the warmer lower elevational limits. So if lower zone specialists shift their ranges 600 meters upslope, as predicted for all biota in the tropics, these species could potentially be confronted with competing species that were more frequently observed in the upper elevation zone. These upper zone specialists, however, will be confronted with the disappearance of their thermal habitat on short mountains. So our results indicate that novel bee communities are likely to be formed in tropical montane landscapes in the future as a result of warming temperatures, and they highlight the urgent need for more work focused on understanding tropical bee ecology, as well as work aimed at developing conservation and mitigation recommendations to support healthy populations of bees as climate change occurs. Okay, so real quick, thank you all for coming again. I want to quickly acknowledge, because I know I'm running short on time here, the San Luis community for allowing us to sample in the area, all the Earthwatch volunteers that helped us collect data, and of course, Dr. Sandra Rehan for helping us develop our Ceratinine reference collection. All right, so
if you guys have any questions, you're welcome to email me here or ask me in the chat, and thank you for coming.